Hello. Today we'll continue our lectures on DNA repair and the mutations. In this lecture, we'll talk about and explain the different mechanisms of DNA repair. As well, we'll describe some of the mutations and give types and the impact as well. And the reference as well for this lecture is Molecular Cell Biology for Ladish et al. And these are the pages that you need to see for the lecture. So what's DNA damage response system? We have a DNA damage response system in every single cell of our body. So this DDR system senses all different types of DNA damage and activates two processes, either DNA repair pathways and cell cycle arrest. So the cell understands that it needs to make a cell cycle arrest once there is a DNA damage response until the damage has been repaired. So the cell cycle arrest can occur either in G1, S, or G2 phases, which depend on whether the DNA damage occurred before the cell cycle entry or during DNA replication. In case of severe DNA damage, rather than attempting to repair the damage, cells undergo apoptosis, which is programmed cell death. One of the key factors of DNA damage response is an important protein, which is actually a transcription factor p53. p53 is a tumor suppressor protein that its normal function is to limit cell proliferation in the face of DNA damage. So basically, once this protein senses any DNA damage, it will limit the proliferation in order not for this DNA damage to be transferred to a daughter cell upon division. So what is B53? B53 is a major protein that involves as well, as we said, in DNA repair, which is the main topic of this lecture. And this DNA repair, you need to understand that it inhibits the initiation of a pre-malignant growth from normal growth. Okay, so it's important to be controlled by B53. As well, B53 controls and promotes three other processes, which is cell cycle arrest, senescence, and apoptosis. And these three processes are very important in the next step in the progression from the pre-malignant growth to the malignant growth and the metastasis. So any abnormality in this this diagrammatic sketch will lead to metastasis in cancer. So here we can see the relation between the DNA damage and DNA repair. There is a lot of damaging agents, including stress to the DNA, oxygen free radicals, ionizing radiations, chemotherapeutics, polyaromatic hydrocarbons, and UV lights. All these DNA damaging agents lead to different damage types, including base mismatches, single-stranded DNA breaks, DNA adducts, which can lead to intra-strand cross-link, as we'll see later in the lecture, as well double-strand DNA breaks, which can lead to inter-strand cross-links. So basically, there are four types of damage, as we said. This base mismatch can may lead to insertions or deletions. The single-strand DNA breaks may lead to abasic sites. The DNA adducts, as I said, may lead to intra-strand cross-linking. And as well, the double-strand break may lead to inter-strand cross-linking. So here, the cells have evolved DNA repair systems to deal with every single type of DNA damage, including mismatch repair, base excision-repair, nucleotide excision-repair, as an example, the transcription-coupled repair and the global genome repair, as well as the double-strand DNA break repair, as the example of it is homologous and non-homologous, homologous recombination, non-homologous end joining repair system. So basically, this figure showed the relation between the damaging agents and the damage type for the DNA, and how the cell repair mechanism deal with every single type of damage. So damage to the DNA is unavoidable and arises in many ways. DNA damage can be caused, as we said, by spontaneous cleavage of chemical bonds in DNA, environmental agents like UV and ionizing radiations, as well as reaction with genotoxic chemicals occur in the environment. Any change in the normal DNA sequence is called a mutation. This mutation can occur during replication when DNA polymerase inserts wrong nucleotide as it reads a damaged template. As well, it can occur at very low frequency as a result of copying errors introduced by the DNA polymerase when they replicate undamaged template. If such a mutation were left uncorrected, the DNA in germ cells might occur too many mutations for viable offspring to be formed. So cellular mechanisms repair damaged DNA and correcting these sequence errors, and this when these repair mechanisms are compromised, the mutation accumulate in the cell's DNA. No surprise that these defects in DNA repair mechanism are main causes of cancer. So let's take a first example and see how DNA polymerase itself can introduce copying errors, but they have the capacity to correct this copying error. So actually the DNA polymerase is considered to be the first line of defense in preventing mutation in DNA by the polymerase itself. As an example, the E. coli, the bacteria, the E. coli DNA polymerase can introduce about 1 incorrect nucleotide every 10,000 polymerized nucleotides. Yet the measured rate in bacterial cells is much lower, about 1 mistake per almost a trillion nucleotide, 10 to the power 9. So why is the difference in this number? Why it's very low, although it can do like 1 nucleotide in 10,000 polymerized nucleotides. This is due to the built-in capacity in the polymerase itself of the bacteria, which is called the proofreading. So actually, here is you can see here is the polymerase, and it's having the template and is building up the, you can see that the light blue is the growing strand, and the dark blue is the template strand. So now, this polymerase is building nucleotides on the template. So the growing end 3' is the one which is growing by the polymerase. So basically, you can see it have 3 fingers, this polymerase, and when incorrect base is incorporated at the 3' end here of the nascent strand, a very good mechanism of proofreading is occurring now, in which, as a result of this, when the polymerase senses that the nucleotide have been inserted wrongly, it moves this 3' end from this polymerase finger here to another finger, another region, another domain, which is have an exonuclease domain. And you can see in this figure, it moves this 3' end to this one. So what happened in this end? In this end, in this domain, these polymerase have an exonuclease activity in which it removes the nucleotide which is wrongly inserted, and then transfer back this 3' end to the polymerization domain for completing the polymerization. So it is correcting itself if it noticed any nucleotide have been inserted into the nascent strand that it is wrong. So what about another type of repair due to the chemical and radiation damage to DNA as well can lead to mutation? So we can see that around 10 to the power 4 or a million per day DNA damage event in a single human cell. Normal cellular reaction, including the movement of electron along the ETC, electron transfer chain in the mitochondria, and the lipid oxidation in peroxisomes, produce several chemicals that damage DNA, including, of course, the hydroxyl radicals and superoxides. These chemicals can also cause mutation that can lead to cancer. Many spontaneous mutations are called point mutations, which actually point mutation, it is a change in a single base pair in a DNA. This point mutation can introduce a stop codon, causing something called nonsense mutation. So nonsense mutation causes a premature termination of protein synthesis. So in other words, this mutation leads to the appearance of a stop codon that normally don't appear. So once the messenger RNA, which will be formed from this DNA sequence, will be shorter and will lead to formation of a truncated version of the normal protein. So nonsense mutation convert a codon which normally encode an amino acid into stop codon. Such a change from UAC as example, which encode tyrosine, to UG that encode stop codon will give rise after translation to a truncated version of a protein that is non-functional. For silent mutation, it doesn't change the amino acid sequence. And if you remember from the lecture of the protein synthesis, you can see, you notice that some amino acids have 6 different codons, like lysine. So it might happen that if there is a pointed mutation in one of the nucleotide, it might lead to the same amino acids, so no major change will be noticed in the protein. Point mutation can also occur in non-protein coding DNA sequence like promoters or enhancers that functions in the regulation of gene transcription. So let's continue about the chemical and radiation damage to DNA, how it can lead to mutation. One of the most frequent cause of point mutation is the deamination of a cytosine base, which converted into uracil. So the process of deamination converts C into U. So you can see here another deamination of modified 5-methyl cytosine. So actually this is 5-methyl cytosine. Upon deamination, it will convert to be a thymine. So these deamination have been found that can lead to, lead to point mutations, as we can see here. You can see here that's a 5-methyl cytosine that's in the wild type, means normally occur and binds with a G. Upon deamination, this methyl cytosine is converted to thymine, to T. If the cells undergo a division and replication of DNA, you will have 2 parent template that will lead to a mutant DNA, because the nascent template now will have an A instead of having G. So this one is leading to a point mutation. So the cells in our body have evolved a base excision repair to repair this modification before the round of replication, in order not to have this point mutation transmitted to the daughter cells. So if these alterations are not corrected before the DNA is replicated, this will create a permanent change in the DNA sequence. So in addition to proofreading, we have cells have Excision-Repair system, ERS, for preventing this mutation, in which a segment of the damaged DNA strand is excised, and the gap is filled by DNA. The gap is filled by the action of the DNA polymerase and ligase as well, using, of course, the complementary DNA strand as a template. So for the base excision repair, it works on the TG mismatches and damaged bases. So you can see in human, the most common type of point mutation is the change of C to a T, as we have just seen in the previous slide, which is caused by deamination of 5-methyl cytosine. The repair system evolved to remove this T and replace it with a C. So you can see here, the damage already happened. The T have been changed by the process of deamination from a methyl cytosine. So a specialized enzyme, which is DNA glycosylase, removes the T, and then another enzyme, which is called AP1 endonuclease, cleaves the single strand. This is followed by AP ligase, which is a part of DNA polymerase beta, that lead to a removal of the segment, the affected segment of DNA. Then with the help of DNA polymerase beta, as well, DNA ligase, it adds again the cytosine to have repaired wild-type DNA. So basically, this explains the base excision repair. Base excision, removal of the defective base, in this case it's the T, and repairing by adding with the help of polymerase beta and the ligase the cytosine. So you can see here this majorly ends up the repair process. You need to keep in mind that the human cells contain a battery of glycosylases. So there are a lot of glycosylases, each of which is specific for a different set of chemically modified DNA bases. So we have different glycosylases for any modified DNA bases. So what about the mismatch excision repair, which deals with other mismatches and small insertion and deletions. So mismatch excision repair mechanism is conserved from bacteria to humans. So this means that it doesn't change through evolution. So basically, it eliminates any base pair mismatch and the insertion or deletion of one or few of nucleotides that are accidentally introduced by DNA polymerase during replication. So in the figure, you can see that here is the DNA, the template strand, and the newly synthesized strand by the DNA polymerase, and these bases have been introduced by this polymerase incorrectly. So once it's sensed, this damage, once it's sensed by the mismatch excision repair, two proteins got attached on this side of DNA, MSH2 and MSH6. And this protein, in collaboration with the endonuclease MLH1 endonuclease, remove, and as well helicase and the exonuclease, remove the whole segment of the single strand of the newly synthesized single strand, and the gap is repaired by the new polymerase and the ligase to make a new repaired segment of DNA in the newly synthesized strand. So in this mismatch excision repair, how this is still unknown, how the mismatch or how these two proteins initially sits on the DNA, it's not known until now how they can differentiate between the template strand and the newly synthesized strand. The mechanism is not known yet. So in contrast to the base excision repair we have just described, the mismatch excision repair occurs after DNA replication. Note, if the MLH1 endonuclease or the MSH2 it was found, or MSH2 gene are non-functional, and this type of mutation is called loss of function mutation. This mismatch repair system is lost, and this may lead to one of the main causes of the inherited form of non-inherited forms of colon cancer. So what about the other type of a DNA repair system, which is the NER, nucleotide excision repair, which deals with chemical adducts, as we said initially, that distort the normal shape of DNA. So cells use this NER to fix some DNA regions contain chemically modified bases, often called chemical adducts. So you can see here an example of a thymine dimer. So here is the normal 2-thiamine residue, T and T, on a single-stranded DNA. For some reason, due to UV light or ionizing radiation, these two T's, T and T, got forming a dimer through a bond formation between these two carbons. This led to a thymine dimer. These dimers have a critical impact on the replication and transcription of DNA. It will halt these processes. Looking into the DNA structure, you can see how the adduct will lead to bulging of the DNA, as you can see here. So there is another set of protein. These protein, they are patrolling and slide along the double-stranded DNA for looking for bulges or other irregularity to send the signal for this NER to work and repair these adducts. Looking closely how the adducts are repaired, so there is important transcription factor which is involved in this process, which is the transcription factor 2H, and is required by the NER in eukaryotic cells because it has an helicase activity that helps to unwind the DNA. So you can see initially the adduct is formed here time in dimer, and it is recognized. Then this transcription factor 2H, one of its unit is XPC, which have an helicase activity. It will melt DNA, opening the complex with the help of the transcription factor H. This opening of the complex and with the help of the endonuclease activity of another set of protein, they will cut the damaged strand of around 24 to 32 bases along the cut is flanking the dimer, the adduct, removing it all this chunk of DNA, and then again the DNA polymerase and the ligase will add the two normal T nucleotides to the damaged part to have again to repair and back again to the wild type DNA. This important use of transcription factor 2H in transcription and the DNA repair, so as we said in our last lecture, that transcription factor, one of the important transcription factors for the transcription of DNA. So it have another function now, it's a DNA repair. And this process is called a transcription coupled repair. So it is very important and explains that DNA damage in higher eukaryote is repaired at a much faster rate in region of the genome being actively transcribed than in non-transcribed regions. So that's to ensure that the messenger RNA which is transcribed having got any mutations from the DNA for proper protein translation. So let's talk about how any damage for the DNA repair system can lead to cancer. So alteration in DNA that lead to the malfunction of tumor-suppressor protein, one of the major tumor-suppressor protein, as we said initially, is the p53. This can lead to oncoprotein. So we have the alteration might be in tumor-suppressor proteins and the production of oncoprotein, which underlie a lot of causes of most cancer. So these oncogenic mutations, these are insertions, deletions, base substitutions, chromosomal amplification, and the translocations through the chromosomes. So the damage to the DNA repair system lead to an increased rate of these genetic alterations. Cells that have accumulating mutations eventually will become cancerous. Tumors have been found to be consisting of heterogeneous population of malignant cells. For this reason, any chemotherapy which is directed toward a single gene or even a group of genes is likely to be ineffective in wiping out all the malignant cells because you have a heterogeneous population of malignant cells. These problems add to the interest in therapies that interfere with the blood supply to tumors. So in other words, therapies have been easier to target the blood supply to the tumor than to target to the gene, because you have a heterogeneous population of malignant cells which have a different set of mutation in every single cells. But all these tumors have a blood supply. So if you aim to target the blood supply, it might be easier to target the tumor. So, but what about the normal dividing cells? The normal dividing cells usually employ several mechanisms to prevent the accumulation of a mutation that can lead to cancer. So in other words, there's a lot of our cells in our body that normally dividing, and even faster dividing or slow dividing. So these kind of cells need to develop another mechanism to prevent the transmission of any mutation to the daughter cells. For example, if multiple mutations are required for a tumor to grow, another mutation for attracting blood supply, another mutation for invading neighboring tissue, another for metastasis, our stem cells, which are very highly dividing cells and have a capacity of replication, is equipped by a low rate of replication combined with normal low rate of mutation, around a trillion wrong nucleotide per single round. So this will provide a shield from being a cancer. Even without the exposure of any external carcinogen or a mutagen, there are some normal, even normal biological processes in our body generate a large amount of DNA damage agents, like the reactive oxygen species, ROS. So let's see here an example how a defective DNA repair system can lead to a cancer. One of the famous examples is the xeroderma pigmentosum, XP. It generally is a heterogeneous autosomal recessive disorder characterized by the increased sensitivity to sunlight with the development of carcinomas at early age. So you can see here, here is the severe case of this condition. So why this type of cancer occur? Actually, they found that these XP genes are seven of the 8 known genes for XP encode component of the excision-repair machinery. So this protein involved in the excision-repair machinery come from gene called XP genes. Actually, from A genes, all of them are called XP genes. They found that 7 of them, once there is a mutation in 7 of these XP genes, this will lead to the absence of this repair mechanism, the excision repair mechanism, which control the cycling of the cells and regulating cell growth and death. So any defect in these 7 genes of the 8 XP genes will lead to xeroderma pigmentosum. So let's see here, what is the difference between a gain of function mutations and the loss of function mutation? Usually gain of function mutation occur in the proto-oncogenes, which will convert it into oncogenes, while the loss of function mutation occur in tumor-suppressor genes, usually are oncogenic. For the gain of function, at least 4 mechanisms can produce oncogenes from their corresponding proto-oncogenes. The first one, of course, is a point mutation. As we said, it is change in single amino acid, a single base pair that result in a hyperactive or constitutively active protein product. The chromosomal translocation that may lead to fusion of 2 genes together to produce a hybrid gene encoding a chimeric protein that is always active, unlike that of the original or the wild type protein. The chromosomal translocation as well can bring a gross regulatory gene under the control of alternative enhancers that cause the inappropriate or overexpression of this gene, which give a gross signal which may lead to a continuous division uncontrolled. While amplification of a gene is a gain of function mutation as well, in which it leads to abnormal DNA replication, in which a DNA segment including a proto-oncogene, so that there will be numerous copies of that gene, leading to overproduction of this encoded protein. So all these 4 mechanisms lead to a gain of function mutation that may convert a proto-oncogene, a gene that normally have a basal level of expression, converting it into an oncogene with a higher level of expression. And these genes actually is concerning with the cell cycle or the progression or the cell division. This will enable cells to undergo a lot of rounds of replication. While the loss of function mutation in tumor suppressor genes, there actually occur among the classes of protein include, of course, of tumor suppressor genes, and they are classified into 5 classes. They either are intracellular protein that regulate or inhibit the entry into the cell cycle, receptor or signal transducers for secreted hormones or developmental signal that inhibit cell proliferation. Checkpoint pathways protein that arrest the cell cycle if DNA is damaged. Protein that promote apoptosis, and enzymes as well that participate in DNA repair. So any loss of function mutation in these 5 classes of proteins will lead to a cancer. So basically this slide shows the difference between the gain of function mutations which convert proto-oncogene into oncogenes, and as well the loss of function of mutations in which the genes which are tumor suppressor genes are turned to be oncogenic. So let's see a perfect example describing how the successive oncogenic mutation can lead to a cancer, and more specifically the colon cancer. So colon cancer, it's a very well-studied cancer and represented a multi-hit model for a cancer, cancer formation from a successive oncogenic mutation. So generally, colon cancer evolved through distinct well-shaped characteristic morphological stages, starting with polyps, then benign adenomas, then carcinomas. So you can see here in the figure, this is a normal colon cells, and we'll talk about what is happening in a section of this mucosal layer of the colon. So initially, the loss of function of an important gene, which is the APC gene, and it's a tumor suppressor gene on chromosome 5, lead to the formation of a polyp, which is outgrowth, small growth on the colon wall. And this polyp starts to grow to form a precancerous tumor growth. And still in this stage, it's not malignant. This APC protein, the APC protein, which is encoded from the APC gene, the tumor suppressor gene, prevents one of the important pathways, which is called the Wnt single transduction pathway, from activating an expression of another gene, which is transcription factor, the c-Myc, a proto-oncogene. And this loss of function of this APC gene leads to the inappropriate production of c-Myc. As we said, the c-Myc is a transcription factor which induces the expression of many genes required for G1S transition of the cell cycle. So again, so what's c-Myc? c-Myc is an important transcription factor that leads to the expression of many genes required for the transition from the G1 to the S cycle, so that the cycling of the cells occur. So ABC gene and the protein product of that genes act as a brake that stops the hyperactivity of these c-Myc genes. Once there is a loss of function of ABC, this c-Myc gene or transcription factor Myc will left uncontrolled. So it will be overexpressed, leading to cycling of the cells uncontrolled. If the mutation in the loss of function of mutation of ABC is followed by activation of another oncogene, which is a KRAS, a gain of function for a KRAS, it's a gene located on chromosome 21, these lead to conversion to a second stage, a class 2 adenomas, which is still benign in that case. Soon after that, it will develop to a class 2 adenomas, class 3 adenomas, which is still benign as well. Once there is a loss of function of one of the major tumor suppressor gene, the one we discussed initially in the lecture, which is B53. Once the loss of B53 in a loss of function mutation occur, and B53 occur on chromosome 17, and it was found that about half of all human tumors carry a loss of function mutation in this crucial gene P53. And this P53, as we said, considered to be a master regulator of the cell cycle, and it is a transcription factor, this P53, one of its main functions. So once this loss of function occur, the class 3 adenomas is converted to a malignant carcinomas, as you can see. So once there is a malignant carcinomas with other changes it either, so you can see here, these the cells multiply uncontrollably, and you can see once they can invade the colon wall, and they can invade to any blood vessel near the colon wall, and then soon they will become, they reach blood supply, and they can metastase all over the body. So to summarize, what are the types of protein that any abnormality in this type of protein can lead to cancer? So generally they are classified into 7 classes of types of protein. The signaling molecule coming from outside the cell, either hormone or growth factor. It could be a protein that is inactive or hyperactive. It's the signal receptor. The second class is the signal receptor, and the receptor is either on the cell membrane or in the cytoplasm and recruited to the nucleus for acting as a transcription, for activating transcription of a certain set of genes. So the second class of proteins is the receptors, again either on the cell membrane or in the cytoplasm. The third set of proteins is the intracellular transducers, which should convey the signal and transfer the signal from the cell surface into the nucleus through process of signaling cascade until the signal reaches the nucleus. Any abnormality in this protein may lead to cancer. The fourth set of proteins that is involved in cancer progression is any transcription factor. We have just two. One majorly the B53, and the second one is the c-MEC. The fifth class of protein is the cell cycle control protein. There is a lot of protein that control the progression through the cell cycle. Any abnormality in this protein may lead to cancer. And as well, majorly, the main topic of our lecture today is the DNA repair protein. Any abnormality in the DNA repair protein may lead to cancer. The seventh class is the apoptotic protein, which deals with and promote the apoptosis of cell in case of the repair system fails. Any abnormality in this apoptotic protein will lead to survival of this damaged cell with its damaged DNA, with its mutated DNA, that soon will be transferred to the daughter cells. So here the slide summarizes 7 different classes of protein that may be involved in cancer. So in this table, we'll see as well, we classify generally into 3 classes. The first class of genes is a proto-oncogenes, which lead to the of cell survival or proliferation. And the example of these gene products are all anti-apoptotic protein component of signaling. Basically, this describes what we have said in the figure. Pathways that result in proliferation as well as transcription factor. And of course, the effect of mutation is a gain of function. And these mutations are genetically dominant and arise by point mutation, chromosomal translocation, or amplification. The second class of genes are the tumor-suppressor gene, which inhibit the survival of or proliferation. And this as well, this actually opposite for the first one, which is apoptosis promoting protein, inhibiting the cell cycle progression and checkpoint pathway, as well as affecting the signaling pathways. And basically, that's the opposite, as we said, it's a loss of function mutation, and they allow unregulated proliferation and survival, and they are generally recessive mutations and arise by deletion, point mutation, and methylation of a DNA. While there are genome maintenance genes, and this of course involved with our topic of the lecture, which is the repair. The main function is the repair or prevent DNA damage. And the gene involved in these cases only the DNA repair enzymes. And of course, the effect on mutation, it's a loss of function mutation because they don't do the job, they don't repair DNA. And which I see better, it's these mutations are genetically recessive. So they only appear once there is a homozygous recessive. And they arise by deletion, point mutation, and methylation. So this table describes the three major classes that implicated in the onset of cancer. So here we'll see how these mutations, these types of mutation, the point mutation, insertion, deletions, and repair of double-strand breaks are involved in cancer. So, and the examples as well. So for this kind of mutation, it can lead to a disease of non-polyposis colorectal cancer, and actually it is actually defective in the DNA mismatch repair mechanism due to the UV or chemical mutagen. And the main type of cancer that may arise is the colon and ovary, and the symptoms it can be detected early because there is early development of tumors. The second example we have just mentioned before, the xeroderma pigmentosum. Actually, it's a defective system is a nucleotide excision-repair, and as well it is the result from a UV irradiation or a point mutation. And it affects skin, it leads to skin carcinomas and melanomas. And the main symptoms for that is skin and eye photosensitivity and keratosis. For the repair of double-strand breaks, you can see the Bloom's syndrome. Actually, the defective system is the double-strand homologous recombination, and the sensitivity or the leading agent is mild alkylating agents. And this kind of defective repair mechanism can lead to carcinomas, leukemias, and lymphomas. The main symptoms is photosensitivity, and as well as chromosomal alterations. The second example is Fanconi anemia. As well, it is the same as the Bloom's syndrome, the defective in the DNA repair system of homologous recombination. Main agents that lead to this kind of anemia is the DNA cross-linking agents. One of the major DNA cross-linking agent is a chemical which is called the ethidium bromide that routinely used in labs of molecular biology for staining DNA. This ethidium bromide have been in agarose gel electrophoresis. This ethidium bromide have been banned in most of the countries due to its severe effect on the chelating with DNA, leading to mutation. The major cancer that may arise is acute myeloid leukemia and squamous cell carcinomas. Of course, these symptoms, you can see it as developmental abnormalities, including anemia and as well infertility. The last example is hereditary breast cancer, which affected two of the important classes of gene, BRCA1, BRCA2, and this due to the defective repair mechanism in the double-strand breaks by homologous recombination. And this actually, it's heredity, so there is no major cause, and it affects breast and ovarian. It can be seen in breast and ovarian cancer, and of course, the symptoms is very obvious and can be detected early. So finally, we can see recently there have been researches on the involvement of the coronavirus with the DNA repair capacity. So you can see here that upon coronavirus infection, it infected different individuals, and this figure represents that the individuals are not the same. There are inter-individual variation towards this exposure, the risk, and the infection susceptibility. So these different individuals have different capacity in their repair mechanism. So not all of them have the same capacity of repairing DNA damage. So this may affect how these individuals will behave upon infection to the corona. This is the latest research. So you can see that there are various outcomes on basis of the repair, the DNA repair capacity of these individuals. Either the infection status may vary, even the treatment outcome, some of the individuals will be treated more efficiently than others, and the recovery and the relapses as well will differ according to the DNA repair capacity in these individuals. Again, these are the reference I've just said in the beginning of the lecture, and these are the pages from the reference you need to read. And these conclude our lecture. I'm waiting for your questions. Thank you.